Okay, so see, here I am going to talk about the sample size calculations procedure in early phase trials. So it's basically towards oncology setup. And next, I would like to present on these survival analysis context sample size calculations. So, like today, Dr. Ashish mentions about different points. Those are required to be kept in mind while we are going for sample size calculations. And before that, we also attended other sessions, and where we understood that there are two types of data, basically the dichotomous data and continuous data. So if you are having continuous data, the brief I want to say, and thereon I will try to start with this topic. So if you see the data types are basically dichotomous or continuous, broadly can be defined as a dichotomous data and continuous data. And similarly, for sample size calculations, all the procedures are based on this type of data only, with the dichotomous data and as well as the continuous data. So if you are having the continuous data, the parameters are measured by mean or median, mostly in mean. In typical in survival analysis, we consider with the median, but mostly in mean if it is a continuous data. And like similarly, if it is a dichotomous or binary data, then we calculate those stuff by proportions. And Ashish already mentioned the way to presentations or for the calculating the sample size towards the proportions, and similarly towards or with these mean. So the means are coming by the type of data you are expecting from your study outcome. So if you are expecting from the study outcome the data is it should be continuous, then obviously you should proceed the sample size calculation with the mean. And if you're expecting your outcomes from your study is coming from the binary way, like proportions, so measurement would be like proportions, and for that Ashish already mentioned the proportions sample size calculations procedure. So these are the two things: type of data and measurement type, and finally the sample size calculations and different formula as he mentions. This is the first step. Second step, let's say what happened and what's ingredient we need to keep it in mind while you are going for sample size calculation. That is another important point. In second level, say, as he mentioned already, see, alpha is required or type 1 is required, beta or power is required or type 2 error is required, and the difference are always required. So difference, that anticipated difference we're expecting, let's say from by deriving the null hypothesis as well as the alternative hypothesis and making the difference between the null and alternative, that will give you the difference. So mostly the prominent ingredient I would say to calculating the sample size is basically the alpha or type 1 error. Both are same way, it's presented in different manner. And similarly the type 1 error alpha, these are fixed, so you are going to fix also the confidence interval. This is also another way, but mostly the type 1 type 2 error and difference. Similarly, if you are dealing with the proportions variable, the way of calculating difference would be the proportion difference, and similarly the type 1 and type 2 error will be fixed similar another way. And if you are dealing with the continuous variable or the mean as a parameters of outcome generations, then you are dealing with the mean difference, and similarly the alpha and beta will be fixed over there. So that's the mostly the second principles of the calculating the sample size. Now thing is that now another important thing, now let's come to point 3. Point 3 is basically the study design. So study design is one of the important point that need to be considered while you are talking about the sample size calculation. So what happened that study design, because study design is nothing but how we are conducting the study, how we are going to collect the data from the patients, or it's not always the patient data. Sometimes we collect from the field also, sometimes we do the survey, sometimes we do cohort study, long follow-up study. So the way we are going to collect the study, conduct the study, that makes sense, or that's having impact on calculating the sample size. So that is need to be keep it in mind where we are calculating sample size calculations for that. So correct. So parameters of way, the parameters we are going to make dichotomous or continuous. Secondly, the ingredient, prominent ingredient, alpha, beta, and difference, and allowing us to make the hypothesis and alternative hypothesis. And third point is the study design we are talking about. So now broadly, if we talk, the broadly study designs can be, if you go with these theory books, and broadly study design can be defined as a cross-sectional study, case control study, cohort study. So basically these are the pillar of study design people start thinking about, and all kind of study designs comes under these broadly comes under these scenarios. Now if you talk about the clinical trials, so that is our main interest of this session. So if you talk about the clinical trials, so in clinical trials we are trying to find out the best effective dose or best promising therapy that is not available in the market, and we want to bring in this market. So that is a basic idea for that. So if we say about what kind of clinical trials design should be, it comes basically as a study design, it comes under the case control study or some extent with this, because in the clinical trial you are keep on following the patients and you have to keep on taking the observations of the patients. So it's also some extent come with the cohort study. But clinical trial altogether, see, they want typical study design, and that study designs is need to have the sample size calculations in different way. But whatever Ashish mentioned until now, same things are going to be applied here, but the way of presentation will be different. The way they're going to make further, that is different. Otherwise, the basic principles are same. What is the basic principle? Basic principle is that if you increase your error rate, if you increase your type 1 error, so error is not always the good point. If you increase your error, your sample size is going to be down. Both the error, if you increase your type 1 error or if you increase your type 2 error, your sample size is going to down. So one principle. Or second thing is that if you make the difference between your anticipated or expected difference between these two treatments, then your sample size is going to be down. If you're having narrow difference, then your sample size is going to be up. So these are the fundamental principles, irrespective of the study design. These principles always work. So as I mentioned, prominent ingredient, and prominent in that ingredient, if you reduce your type 1 error, obviously your sample size is going to be up. If you increase your type 1 error, your sample size is going to be down. If you increase your type 2 error, sample size is going to be down. If you reduce your type 2 error, sample size is going to be up. If you reduce your difference, sample size is going to be up. If you increase your difference, sample size is going to be down. So that is the fundamental principle. Now let's come to this clinical trial prospect that we are going to talk. In oncology, as you say that broadly this study design can be, or oncological trial can be separated into phase 1, phase 2, phase 3, and phase 4. I'm not going to talk about the phase 4. Phase 4 is very rarely done, and phase 4 is mostly, I would say, it's a real world evidence data, so you don't need too much worried about this conducting a proper trial, because people are already having data and people just find and monitor what's going to happen over there. So, and it's a post-marketing surveillance altogether, so you don't need to worry or to calculating a sample size formula and other stuff for phase 4 study. So leave that one. Now let's come the phase 1 to phase 3. What's happened with this phase 1, 2, and 3? As these phases are separated, so in that sense, there are way of handling the data, way of conducting the study, and simultaneously the sample size calculations scenarios are keep on changing. The things are different. So now I'm going to talk about this phase 1 first, or sometimes I will touch in this topic phase 2 as well, because phase 1 and phase 2 are sometimes called as early phase trial. So here I'm going to touch mostly on the phase 1 and phase 2 trial objectives, or a way of calculating the sample size. Your scenario need to be kept in mind while you are going to calculate the sample size. And in my next presentations, I'm going to make the phase 2, phase 3 studies, or that typically the survival analysis context, I would say. Now let's say here the phase, and it's very thin line to make difference phase 2 and phase 3. Sometimes after having so many years of experience, having so many trials, still we get confused. The study should be put into phase 2 or phase 3 if it is a approved trial. I'm not now talking about two types of drugs, or in terms of the in the clinical trial sample size calculations. It's becomes very easy to present the work if you consider that the molecules or the medical drugs are there. But apart from that, in radiation oncology also there is a different gray doses are there. That stuff also works simultaneously in the similar way here. But unfortunately, the surgical oncology part we don't have, because it's a quite open level. We don't have the blinding part and all this stuff. So better to avoid these surgical oncology part here. So better if you can consider on the medical oncology or radiation oncology, because every time these two domain deal with the different doses and different combinations of drug. Now, if you talk about this academic institutes, we mostly work with these approved drugs, but we try to make between different shifting of the doses level and trying to find out which doses work better. But if you talk about the industry, like Pfizer's and other pharmaceutical company, they work with a typical new molecules, and here it's quite sensitive to establish the new molecules and achieve the best effective drug over there. So that's a very huge cost involvement as well over there. So, but more or less, as far as the study design context, both things, if you talk about the new molecules or the existing molecule with different, different combinations of the drug administrations, in both cases, these phases or study designs are similar. Only we have to look how it's going on. So now, let's say conventional paradigm. The conventional clinical trials are evaluating the effect of a new agents or agents in human has three phases that I already mentioned, that phase 1, phase 2, and phase 3. So the phase 1 basically goal is to determine the acceptability of the safe dose based on the toxicity. For a single agent, a set of doses to be explored in specified manner. If two agents given in a combinations, or like say combined therapy, are to be studied, and a set of those are paired to be specified either as a rectangular grid or staircase shapes, where only a two doses increase each step. So what does it mean? It does mean that we are much more, while you are talking about phase 1 trial, irrespective of the new molecule or agent or existing molecule with different drug administrations procedure. In both the cases, we are much more concerned about the toxicity rather than efficacy. Okay, so here nobody in phase 1 trial, nobody is going to talk about the efficacy. We are not concerned about the efficacy. We know that since it's a new molecule, there must be some kind of efficacy must be over there, but we're not at all interested to look about the efficacy. Here we only concerned about the toxicity and how much toxicity can be adjusted or can be tolerable by a patient. So that's about main concerns. And based on that, sample size calculations procedure for phase 1 trial is also existing in that manner. So here we look on the toxicity level, and based on that we decide that and which doses or drug should be taken further. However, in some phase 1 trials, doses are chosen from a continuum, either an interval for an agent or rectangular set of pair agents. And for that, Peter Thal from MD Anderson's, he devoted his entire career. He started his career in '92, and until now, this fellow, genius fellow, is keep on working on this phase 1 trial. If you go and Google with the MD Anderson's website with the Peter Thal, you will get several articles. He worked on the phase 1 clinical trial, toxicity and efficacy, regime define, and all this stuff. So phase 1 is conducted by the treatment small cohort of the patients, most commonly with size 3, 2, or 1. Why? Because as I mentioned that here we are much more concerned about the toxicity. So we don't allow so many number of patients to take at all at a time. So we started with a very small step. We take only 1 patient, 2 patient, 3 patients, and see if things going well or not. Then we pursue further and keep on taking more number of patients. So that is not the conventional way of the sample size calculation. So conventional way of sample size calculation, always keep it in mind that how many number of patients you need until we will conduct study and like that. But here, toxicity is a toxicity, very much sensitive issue. We have to go very cautiously, and 1, 2, and 3 patients we start and see what's going on, and after that we proceed on that when we are going to make a prescribe that dose is useful or not. So doses or pair of doses. Now thing is that, is while you are talking about the doses, you can say the single doses or monotherapy, or you can say combined therapy, or different administrative rules can also play over there. And the doses, dose pairs, each successive cohort is chosen adaptively based on the dose toxicities that is being observed on the previous cohort. So it is not, you have to start the study somewhere. It's not that always you should have the data, but as far as suitable data, if you are having in other studies or earlier trial, that can be taken further, so that can help you some extent to decide your starting point of dose. It's not that somebody in a cisplatin is going to have with, or particular drug is going to have with this, let's say 250 mg, and suddenly you started to think, no, I want to do a conduct a study with 2500 mg. That's not going to have, because you have to rely with some near to the 250 mg, because that's people are keep on giving. So suddenly we can jump with the new doses and try to find out sample size or conduct a study. That's not going to happen. The most important part for these early phase trial is the MTD, maximum tolerance dose. It's defined as, depending on the method you use, each cohort dose may be above, below, or the same as that the previous cohort. The final chosen dose is often called as a maximum tolerance dose. Conventional phase 1 design don't use efficacy in dose finding algorithm, but usually record is a secondary outcome. That I mentioned, that we never bother about the efficacy of a new drug in phase 1 trial. We always concerned about the toxicity of a trial. And while you are talking about the toxicity, the principle we consider here is that if you give more doses, tumor will be, I am talking about the cancer or the tumor. So basically the tumor will be under control. If persons, the general understanding or principle is that if persons can tolerate 2500 mg, give it to them, it will be under control. Rather than 2500, if you can tolerate 5000 mg, give it to them. So the general fundamental principle is that as much as dose you will give, person's tumor will be under control. That is the general principle. But that's the way the drug work over there in the body. That people assume. But simultaneously, you have to keep rush on that, and you have to put that, no, you can't give any kind of dose, because it should not be more than tolerable dose of a person. So that's the MTD stand over there. So now how the general principles work. If you talk about that, even in conventional trial also we have seen, if you keep on increasing, so if you see my x-axis is stating about the doses, and if you keep on increasing the doses, it says that the blue line and red line. So red line is basically you are keep on increasing the doses, and the tolerable limit is going to be reached. And simultaneously, if you see that some extent, the blue line is nothing but the efficacy. So some extent, efficacy gets optimal. After that, it also drop down. So it's nothing that if you keep on increasing doses, you are going to get the best results. No, that never happened. So after certain time, after giving certain doses of a drug, person's efficacy get optimal level and it started to getting down over there. So that's the way. So we have to find out what's the best dose we can deliver, and that is need to be considered. So, and based on that we perform. So here we don't have like earlier studies, earlier presentations, Dr. Ashish mentioned that what should be the sample size or derived sample size. But here we don't have the prior sample size. We have to conduct the trial in continuous manner, and we have to get the results, and based on the number of persons, and we have to put the evidence on it. Now comes to the metronomic therapy and efficacy that because since I worked Tata Memorial Centers and I'm fortunate to work with a new way of drug administration. So that is the metronomic chemotherapy. So here whatever the literature you will get, as I mentioned, that phase 1 trial, sample size calculations, that Peter Thal and others, they work on that. Those are basically the principle that if you give more doses, person's drug or tumor will be under control, and that is the MTD. But while I worked here, that's it. The new way of looking things comes here. That is called metronomic chemotherapy. Here, what happened that we have to do the phase 1 trial because we don't know what the particular doses of a drug should be, but simultaneously doses are very minimal doses, very low doses of that. So if you are having already market approved doses, let's say 500 mg can be given to a patient based on his BMI and others prognostic status. If it says that market approved drug says that it is a 500 mg could be given to a patient. But here in Tata, we started to working on that. No, out of 500 mg, we can give only the 50 mg. But the drug administration should be different. 500 mg given as a 21-day cycle, but 50 mg would be given continuous manner for 6 months. Since 50 mg we are giving, there is no chance to having the toxicity because you are already 1/10 of the existing or the approved dose. So don't need to be concerned about the toxicity. But in the phase 1 paradigm, we have to show that yes, 50 mg is always is acceptable toxicity as well. But then how should we proceed? Next questions comes to us. Then if you talk about the 50 mg, should we go for 50 mg or we should go for 100 mg or we should go for 20 mg or we should go 150 mg? Still, out of 500, 50, 20, 100, 150, all these are very safest dose. But still, which one we are going to make? So in that context, so we started to getting that thing, okay, which things, since we are having the toxicity is under control, or we can expect the toxicity will be under control, then we can think about the efficacy. But people never talk about the efficacy earlier in the phase 1 trial. People only talk about the toxicity in this trial. So while we are starting to thinking about the phase 1 trial and as well as the deriving that efficacy and giving priority to the efficacy than toxicity, then life become little challenging. And then we derived new formula, new methods, and new way to calculating the sample size and find out this, let's say 20 mg, 50 mg, 100 mg, 150 mg, which one is the best dose. So based on that, we only consider the different kind of biomarkers available, and based on that, we try to find out which one is the best dose. And fortunately, these papers got, based on these reasons, the paper got published in The Lancet, and if you want have, you can go into details on this paper and you see how the metronomic dose or low dose chemotherapy dose defined can be done. But it's a different way to work on that. It's not the toxicity. So the first time we have seen that toxicity can be avoided to look on the phase 1 trial sample size calculations, and we can do it in a different manner. Okay. So now I'm going to for this again. I'm coming for the phase 1, the typical conventional way, because whatever I mentioned, it's a one challenge we faced. Obviously, if you want to have interest on that, metronomic chemotherapy, where efficacy is can be given much more value than toxicity because we are free from the toxicity. We can go in that, but let's say come to the conventional approach with this MTD and phase 1 trial. what says. So phase 1 trial, so also called as an expansion of the cohort of some specified number of additional patients in the treated group as chosen as the MTD in order to obtain a more reliable estimator of the probability of toxicity at that dose. So it's a basically, it's an ongoing process. You don't know how, you don't need to specify the sample size. You started with some value, started with 1, 2, 3 patients and keep on going and try to find out. Just give you one patient, one dose. Let's see what's happened. The dose is tolerable or not. If it is a tolerable, go to next patient, give the same dose, see it. And then if it is okay, then go to next patients and see it is again okay or not. Like that, the way it's worked. So you don't need to have, you don't need to promise that 30, 40, 70 number of patients you are going to have in your trial. Obviously, phase 1 trial, if you see the literature, so book, here you will see phase 1 trial should be done with the 30, 40 patients. But it does not mean that you have to specify the number. You can keep on going the trial and end the trial with 30, 40 patients number. So underlying motivation is about the assumption that dose is increase, the probability of the both toxicity and efficacy must increase. That I mentioned that in the conventional MTD trial, that it is always that the efficacy and toxicity will increase if you increase the dose. Since efficacy is not used for making decision in the conventional phase 1 trial, logical flows to undesirable consequences of making this implicit assumption and determining the MTD based on toxicity. Once an MTD has been determined in phase 1, conventional treatment efficacy is evaluated in phase 2. So the way it works like that, the phase 1 is only devoted for calculating the or obtain the best dose, that tolerable dose that is under control with the toxicity, and take forward that dose to prove how much it is effective into phase 2 trial. So most phase 2 designs are based on the binary indicator of the efficacy with the secondary aim to estimate the variety of the outcomes such as pharmacokinetic or pharmacodynamic variables. The rationale of using yearly efficacy along for phase 2 treatment evaluations has 3 components. The first component is that to increase the probability of long-term patients' benefits, such as existing survival time. Okay, so until now, what I understood, or we understood that phase 1 is basically talk about the toxicity. Now, if you come to the phase 2 is now concerned about the efficacy. The primary challenge we face while you conduct the phase 2 trial is that efficacy I'm talking about the oncology setup, is that it takes long time to find the efficacy. Efficacy, what do you say? What if you say that what efficacy means in oncology, patients having the prolonged survival, right? Having long-term survival. So if you talk about that sense, so how many days, months, or year we have to wait to find out the best doses? That's a big challenge. And like phase 1, it's immediately result you will get, because today you are giving dose, tomorrow, day after tomorrow, or within 7 days, you see the persons are having fever, vomiting, other toxicity impact or not, and immediately you can document that, okay, this dose is not effective or this dose is effective because persons have more toxicity, don't allow them. So that is a very immediately you can get the results for phase 1 toxicity understanding or evaluations. But in case of phase 2, while you are shifting our objective from toxicity to efficacy, that becomes very challenging because we don't have the immediate outcome. We have to wait a long. Nothing wrong to wait for long term, but problem is that once you are having 4 or 5 doses or choice, like as I mentioned in phase 1, we are having 20 mg, 50 mg, 100 mg, 150 mg. So 3-4 choices are there, and immediately by looking at toxicity, you can find out that which one is the best. But while you are talking about the phase 2, then you can't proceed with 4 and 5 options of the doses and take that which one is the best one. That is not possible to run a trial for long run. So that is the fundamental challenge to get the earlier results in phase 2 trial. but it is always expected to get the long-run patient benefit. Secondly, the earlier treatment evolution is often impractical to wait to observe long-term event that I mentioned, and earlier treatment evolutions, you are not going to give it that, but that is expected. And thirdly, since MTD has been determined in phase 1, safety is no longer major concern. So in phase 2, we are not bothered about the toxicity, because we already established that this drug is very much toxic free. So people can very much tolerate that dose. So proceed with that. So thankfully, phase 2, we don't need to worry about the toxicity. However, as far as GCP and other things are concerned, if you have to monitor cautiously, you have to check the toxicity event over there, and you have to document it properly. But it's a secondary thing. As far as the study design, sample size, objectives are concerned, this is not the primary thing. In oncology, most phase 2 trials of new experiment treatments are single arm without randomizations against a standard arm. While randomized phase 2 trials are used more commonly in other disease areas, in oncology, a culture is basically the single arm phase 2 trial continues to persist. So that is the thing, but it's not that we don't do any phase 2 trial in oncology with double regime or 2 arms. It happens, but mostly we'd say that it's basically the single arm study. Now come how to do with this one. The procedure or calculating the sample size. As I mentioned, there is in early phase trial, there is not any simplest approach to calculate the sample size in early phase. The way you are going to conduct the study, that is important for phase 1, and that's merged with the sample size here. One of the technique is basically called as a continual reassessment method. As I mentioned, the scenario is like, you are keep on observing the patients, give a patients, look on the toxicity, and keep on proceeding and keep on giving the same dosage again and again. The continuous reassessment methods, it's coined Quigley et al. In 1990, is a model-based sequential adaptive phase 1 dose finding design. The CRM was the first design of its kind proposed to dose finding in early phase trials, but because it uses toxicity but not the efficacy. What is the assumptions behind the CRM? CRM assumes a paired dose toxicity curves. So and repeatedly updates these curves using the observed accumulated toxicity outcome from the patients in the trial. Based on the updated dose toxicity curve, each new cohort of patients is assigned to the dose having posterior mean of toxicity probability closest to a pre-specified fixed targeted toxicity probability. What does it mean? So we are having a typical kind of cards are there, and we are keep on accumulating patients over there by looking their probability of toxicity and success, and we try to find out that curves is going well or not, and it is based on that our desired expected results or not. If it is wrong, then we should stop the study or adaptive study, or stop the study and finalize the doses. Otherwise, keep on finding the best dose by looking at toxicity. Because the CRM uses the observed dose toxicity data, assume a dose toxicity model and has explicit criteria for selecting doses has in an impact, and it's triggered the subsequent development of numerous sequential adaptive design for early phase trial. So based on these approaches, based on the CRM methods, later on the sequential adaptive designs were developed. So, and one of the CRM methods, I would say that is a very straightforward methods and very approachable, and people can easily adopt in any trials, irrespective if they want to have that company approved or the market approved drugs. Still, you want to see that among them their patients, just for this sake of understanding or doing the experimental or exercise they want to do, they can easily conduct the 3+3 algorithm or 3+3 study design. For a phase 1, the CRM is much more efficient and yields substantially better operating characteristics than so-called 3+3 design. So I'm now going for this, how these 3+3 designs happen. So now let's say MTD is the highest dose, as we mentioned, at which no more than 1 out of 6 evaluated patients has to had a DLT. Now I'm using the new term called DLT. I'm coming, what is a DLT? So 6 patients should be treated before the dose is declared as MTD. So maximum tolerable dose is basically that I mentioned that people can having, and but simultaneously where the doses we should stop that dose, and we should not proceed further, we should not increase the dose, and that is called that DLT. So basically these works in a two-way, this finding the best dose by escalating or deescalating, and the principle is that as we were trying to always find out the MTD, maximum tolerance dose, the highest dose, so we always started with the accelerating modes. Unless and until it's very much required, we don't go for decelerating road, always we go for escalating road. So what's happened here, we started with 6 patients only, as I mentioned, because we are talking about the new doses, or you don't know, and you should be having very much sensitive about this dose choosing. So we started with the 6 people, or 6 patients, and we out of that, and we give one of them give the particular dose and try to find out what's going to happen. So how this, and simultaneously we can roll the patients, we can one patient's results comes that we expect that toxicity result can come within 7 days. The next patient's recruited, again go for 7 days, let's check, and let's go for third patient, like that it's happened. So what happened, we choose first that here is the 2 things here, the patients with the DLT at a given time, and enter 3 patients at next dose level. So out of 3 patients, if we don't have anybody is having toxicity, okay, so we are safe. So that's mean the doses we consider that is safe, and persons can be given that dose. So if 0/3, 0 means how many were having raised with the toxicity. If none of them having raised with the toxicity, that the dose is very safe, at least safe, so we can go for the next accelerating procedure for. So let's say we started with the 220 mg. Now we consider only the 3 patients and find out of 3 none of them having toxicity. Then we consider for 50 patients, we go 50 mg, we can go for 50 mg dose regime, because we are going for 1-step mode. So at least what happened, if the next step, if 0 or of these patients experience DLT, then that is fine, good, we can go for next level. In next level load, that is I was talking about the 50 mg, out of these 50 mg, the 3 patients given, if 1 or more than 1 patients are having toxicity, then what is going to happen, if more than 2 patients are going to having toxicity, then what is going to happen. So particular doses, we are going to have 3%, and 3%, 3 things can happen. Either no one will be having toxicity, secondly, one of them could be having toxicity, and thirdly, 2 or more than 2, means all of them would be having toxicity. If 0, if none of them having toxicity, then we can proceed for the next dose level. If one of them are having toxicity, then the scenario is that wait, recruit more 3 patients from there. So initially we are having 3 patients, and out of that 1 is having toxicity. Then in the same doses like 220 mg, you recruit more 3 patients, see what happened. If out of 6, total out of 6, because 3 earlier, and out of them 3 only 1 is having toxicity, and next 3, and out of them none of them having toxicity, so 1 out of 6 is okay. So you can go for next dose level, you can accelerate the dose. If it is happy 2 out of 6 are having toxicity, then you should stop here, and you should keep on recruit 3 more patients and try to see what happened. If again out of these 3 are having, let's say none of them having any toxicity, so 2 out of 9 is going to having toxicity, so you can go for next dose level. But if 3 out of 9 is having toxicity, So you should stop and come to the below level. So that is the way. Come to 20. But here from 50, you can go for 100 or not. It's depend if out of this, the patients given the 50 mg, among those 6 patients, only one is having toxicity, then this procedure or principle allow you can go for the 100 mg dose. And again, you can record, more 3 patients and see what's happening. So what happened, basically, you are keep on recruiting new patients in new regime, or if it is a quite sensitive, or you think there is a alarming situations to go to next doses because more number of patients are having the toxicity. So you should retain that dose. You try to find out, bring more patients in that specific dose, and convince yourself that okay, this is okay, we can go for next level or not level. So that's the general principle. Now, these things are basically, we are talking about the toxicity. Ultimately, these are going to come to a percentage, and the percentage is the way calculating the sample size Ashish has mentioned, but I'm not going to bring that sample size calculations here, but the principle works like that only. Okay, so general rules: never re-escalate to a level after the de-escalating from that level. That means you started with the 20 mg, go to 50 mg, and then come down to 20 mg, and then don't go again for the 50 mg. That's stop us to go for. So you can't go and come. You can't know. If you one time you can go to accelerate, and one time you can de-escalate, but again you can't accelerate. If the decision is to de-escalate or choosing one level lower, but the current level is the lowest, stop the trial and choose no level. So basically, it's not giving you the chance to consider the dose or drug or experiment at all. Okay. Thirdly, if the decision is to escalate about the highest level, stop and choose no level. So if the decision is to escalate about the highest level, so basically, the 50 mg you given, and out of 50 mg, 9 patients treated, and out of them, none of them are having any toxicity. So basically, the decision saying from 50 mg you can go to 100 mg. Correct. But above the highest level, but it's not that you should go 100 mg unless and until it is proven. 100 mg is proven, as I said. So it is better to consider retain with this 50 mg. And the decision is to stop and choose the one level lower, but one level lower has 3 or fewer patients, treat more at that lower level. So now, as I mentioned, so you started with the 20 mg, you took only the 3 patients, and out of that, none of them are having toxicity. That's why you go for this, one step more. But you find that something is wrong over there in the 50 mg, you come down to 20 mg and found that, but to conclude that 20 is safe enough, you have to take more number of patients in the 20 mg region. So this is the flow chart. What start at the prescribed starting dose, treat a patient or a cohort of a patients, reach the maximum sample size. If you reach it, then stop the trial and select as a MTD. If not, then compute the DLT rate at the current dose. If it is a more than 35%, so basically how it is going to be have the 35%, so 0.359, basically that, as I mentioned, that 1 out of 3, that is basically around 33% happen, but 33 with standard deviations, it comes around 35%, and 33 minus standard deviation, it comes around 23.6, something like that. So that's a two-way it goes. So if you compute the DLT rate at the current dose, if it is a more than 35%, de-escalate the dose. So these doses are not going to be tolerated, stop it, goes down to the low level. And if it is a tolerable, escalate it and proceed with that. And it is always expected that with the range should be between 0.236 to 0.359. So this is not the arbitrary value. It's not that you can't decide your own. It's a standard value, and you have to follow that rule. And based on several simulation studies, these values are being established. So we have to follow this rule, and there is nothing we can't change that value. So, and then retain the current dose over there. So the DLT is defined as the total number of patients who experience DLT at current dose divided by the total number of available patients treated at the current dose. Okay, so now comes how to do. Before that, I would say that CRMs, for that there are other software also. If you go to the Peter Thol's website, they will get so many packages are there available. 3+3 designs softwares are available, and these are very standard, because I would say, because they are the authors of these papers, and many commercial or proprietary softwares don't have that capacity, that power to calculate the phase 1 trial. I'm talking about the phase 1 trial sample size calculation. So then these people, these guys, because you can say that I want to work with the SAS, I want to with these so many softwares to calculate the early phase trial, but I would say go to their website. There are so many small apps they put over there. You can use this blindly because they are the authors. They prepare their packages, and those are mostly effective than any proprietary software. Now, I'm coming for this one another way that most approachable software is that R, and R is the best, I would say, because as far as the computations and this advancement of the statistics things are concerned. So that's why there are some way of this, but thing is that there is a principle behind that very rare people do explain on that about the principle of to calculating the sample size that I mentioned, that calculating sample size with the way it should be calculated. So basically, until now we all talked about the assumptions and the hypothesis and other part, but as I mentioned, as a MTD and the metronomic chemotherapy things I mentioned. So what happened? The way it need to be calculated, there is a two-way. One thing is obviously the Bayesian way to calculate the sample size, and another thing is the conventional likelihood approach way. So 3+3 design or CRM or MTD design, mostly these are based on the maximum likelihood approach or the frequentist approach. But if you want to go further on that, and if you are much more interested on that, to look on the phase 1 trial sample size calculation, so there is another way to calculate, that is called Bayesian approach also. And Bayesian optimal doses is one of the way to calculate. So it says that what should be the optimal value. So until now we are talking about that 20, 50, 100, 150, but the optimal value can be decided some extent by this package. And this package is library called BOIN is available, and you can install it in R, or you can go to YouTube and check how to install R and packages, and can say, and it says only this get boundary value and targeted value. You have to specify only number of cohort, or means what I mean, if you how many times you want to conduct the study. So if you want to conduct the different repetition of the study, like escalating, de-escalating steps several times, so you can go for the number of cohort as 10. Just it's given as a n cohort is equal to 10, and cohort size every time you can give as a 3. So 3 is a, by default, it is a best approach, best number to be considered. So we will go that you will get the summary statistics, and you will get that results to go for DLT or not. So obviously DLT always allow us that not choose. You should have that escalate dose if the observed DLT rate is below 23%, de-escalate dose if the observed DLT current dose is more than 35%, and it shows that entered chain. So as I mentioned, 10 cohort size you should mention. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So here are 10 cohorts are been mentioned here, and each cohort we are going to consider the 3 patients each time, 3, 6, 9, 12, like that. And it says that escalate if for each cohort you are going to get 0, 1, 2, 3, like this number of patients. So then if out of 3% only 0% are having no toxicity or DLT, then you can go for accelerating like 6 out of 6, only one is having toxicity, then you can accelerate. Similarly, out of 30, it's a cumulative number. Out of 30, if 7s are having toxicity, then you can go for the next level. Similarly, it says that they're decelerating. How to? If out of 3, initially 2% are having toxicity, you should decelerate and go to lower dose, lower drug dose, like 3, similarly, and like this, all things going to happen. And eliminate, if means you should not proceed at all, neither escalating nor de-escalating. If out of 3 is having, 3, all of them having toxicity. Out of 6, 4 are having toxicity. Out of 9, 5 are having toxicity. Then you should not consider the dose at all and completely discard that thing. So this is the simplest understanding. So if this is the principles or results, so you are in one place, you are having these outcomes, and this is a standard outcome, irrespective of the trial, irrespective of the study design, it stands properly here. And in another part, you are conducting this trial. So like I said, that here you don't need to consider any typical trial study designs or sample size. This is a principle. You can put this thing note here, and you can conduct a study, and you can take a call while you are continuously conducting the study and see what's going to happen. And now, it's most rigorously given. So this the entire part is again elaborated with the patient to patient, while we are keep on, rather than groupified, rather than again each cohort side or each dose level or experiment, the entire things, because this is the way the entire study is going to happen. Every time, as I mentioned, it's quite sensitive. So every time you are going to take a new patients and going to look about the toxicity. So every time you are going to take a new patients, look about the toxicity, calculate the toxicity, and take a decisions on that. So this is the way to find out the dose accelerating procedure using R. And this another steps, if you can make, if you want to have that, what's the doses should be regime, like 0.5, 0.05, 0.15, 0.3, 0.4, 5.6. So that can be also be obtained from that results. And with this average estimation percentage on that. So it gives us selecting the percentage at each dose level, and average number of patients should be treated for particular dose level, and average number of toxicity observed each dose level. So it gives more depth about that way, but I would say the best way approach is to start with this earlier code and see and put it in handy, and you can proceed further. These are the 3+3 algorithms again. So 3+3 algorithms, once you're conducting the 3+3 algorithms, generally you can see the particular dose level, how many people are raised with the toxicity, and that percentage or that number you can present, and you can take a call. Similarly, you can take that into that number, you can convert into percentage, and again you can it would help you to take a call. And number of patients allocated, so that is also important, because how many patients are allocated particular doses, that you can take a call by presenting this simple histograms on that. Now, phase 2 cancer trial, most commonly Simon's two-stage designs works nicely, is prepared in 1989. And the design include an earlier stopping rule after the first dose, first stage, I would say. It has two versions. One is optimized to minimize the expected sample size, and the other is to minimize the maximum sample size, each subject to specified with the type 1 and type 2 error. So now see, phase 1 is typically the toxicity involvement, but phase 2 is bringing with this toxicity as well as the efficacy. But here, efficacy taking the leading role here. So I efficacy taking leading role important. So what happened here? We are much more towards the Ashish mentioned that sample size calculations deriving the procedures. So here we are having some kind of expected value, some kind of null hypothesis, alternative hypothesis, and we can derive the sample size here prior to our study, and we can achieve and based on that derived sample size. But there are some challenges There are numerous other phase 2 designs are there, and they are very large. Literature exists. More complex phase 2 designs mostly include more than one interim analysis. So while you are talking about the phase 2 design, so now things are concerned that, how many years should I consider? Should we conduct the study? 5 years, 6 years, 7 years, 9 years? And it's worthy to consider that 5 years, 7 years, or 9 years study or not. So should we stop the study, and should we look at the study after 2 years or 3 years? So that's the reason we have to have some interim analysis, look on the data, check that whatever we assumed or expected, that is going well or not. So now in the phase 2 study, the same kind of efficacy would be assessed after a couple of months or a couple of years, while your study is several years already mentioned. So in between, you want to assess the study, that's give the rise of the interim analysis. And once you are talking about the interim analysis, placing the interim analysis, it should be in a 6 months, 1 year, 2 years. That's make an impact that's make a direct impact on the study design. And while the study design is having impact, obviously sample size calculation is also going to be impacted. There are different way alpha spending functions are there and to handle those things. But these are the existing challenge are there. Delayed efficacy. So sometimes, some sites of cancer efficacy can be measured after a long time. Obviously, there are two ways we consider the date. If you consider the date as a survival or the efficacy outcome, obviously that is going to take some extent prolonged time. Not all cancer site, but some cancer site. But that's why we shift to some date or as a survival to some progressions as a survivals. I will come to the next slide on that, next sessions on that. So that's delayed efficacy and others monitoring the efficacy and toxicity. And I would say that there is ample work been done on that. You can see, and if you can interest on that, and mostly these packages are now available and linked with your work. While randomization is more common in phase 2 trial outside oncology, the use of randomization is very limited in phase 2 cancer trial. How Simon's two-stage works? Simon design is very simple and very easy to implement using freely available computer program. It allows one to specify the fixed null and alternative response rate or probability terms and type 1 and type 2 error probability. And given these 4 inputs value, quickly to compute the sample size and decisions cutoff for the 2 stage. In phase 2, oncology setting where multiple experiment treatment must be screened, Simon et al. Proposed the idea of using randomization in phase 2 clinical trial to obtain the unbiased treatment comparison. Unfortunately, despite several years, randomization in phase 2 trial remain controversial. So how to conduct the Simon's two-stage design? So again, you need to have few ingredient or prominent ingredient that Ashish already mentioned. So obviously, type 1 error and type 2 error rate is required. In addition to that, response rate or probability of success or proportions. So all these terms are same. These things are required. So here we can say that unacceptable response rate. Here we can say response rate that is desirable. So these 2 points PU and PA, this is nothing but are going to give you these 2 proportions, as well as in the function itself, it will calculate the difference value. So difference, or that he mentioned that D can be calculated from here. And EPI and EP1 and EP2, these are basically the threshold for probability of declaring drug desirable under P0 and threshold for probability of rejecting the drug under P1. And maximum total sample size can be obtained based on that. So you see here, this simple only one line code is available, and you need to run only the PH2 Simon with 0.2, 0.5, 0.1 and 0.1. So 0.2 as your unacceptable. So you're not going to say that if my response rate is below 20, you are not going to allow the drug. So you're talking about the efficacy. So if you are 100 patients are treating, so out of that 20 patients and above would be having response on that. So that's why 0.2 you are going to consider here, and 0.4 you are going to consider here, because response rate you are expecting. So you are not going to You are not going to throw the study if it is more than 6.6 or more than 0.4, because it's a very positive remarks, positive things you are going to get. But you are going to throw the study if it is a less than 0.2. So basically the boundary you are making as a at least 0.2, and you are expecting to 0.4. That's two things are required. In addition to that, that type 1 and type 2. So if you make the type 1 and alpha value and type 2, so you are always going to get the alpha 0.5 and beta as alpha as 0.1 and beta as 0.1, and optimum value and minimax value are going to get, and it's land you with the sample size as a 37 or 36. So since again phase 2 trials is having small sample size as well as compared to phase 3 study, but it will land with 37 or 36 a number of patients. So mostly these are the sample size calculations formula for phase 1 and phase 2. Now having the sequential and other study design, things are very rigorously we need to look on that. But I would suggest for starting point, these two methods like Simon's methods and 3+3 methods could be one way. For phase 1, 3+3 is the best one, and phase 2 Simon's method is the best one. With that note, I would like to end it here. Now, say any comments or any questions are there?